Hello, here's the second part of Lab 6 Passive Transport, which would be Osmosis, by Victoria and I. So the introduction, first of all, what is osmosis? And osmosis is the diffusion of water across a membrane. It's used to balance solute concentrations when a solute cannot move across the membrane itself. And like diffusion, it is spontaneous and does not require energy. Different solute concentrations have different effects. When a solution is isotonic, it has the same amount of solute as the inside of the cell. And when you see this, the cell is shaped like it should be. There's no problems. When a solution is hypertonic, it has a greater concentration of solutes than the cell. So the cell crenates or shrinks or shrivels. And when a solution is hypotonic, it has less of a concentration of solutes than the cell, and the cell bursts or lyses. So here are some pictures of different concentrations in osmosis. This is isotonic, where it's equal. Hypertonic, where the solute concentration outside the cell is greater, and hypotonic, where the inside of the cell has a greater concentration. So for our experiment for osmosis, well, there were two parts. The first was how osmosis affects the concentrations of different solutions, and then how osmosis affects cells. So for the first experiment, which was how osmosis affects different solutions, our hypothesis was that water will move faster into the dialysis bags, or tubes, containing the higher concentration of sucrose. Just going back a second, the dialysis tubes are used to replace plasma membranes. They are semi-permeable. They allow water to pass through, but in this case, not sucrose. And the bags with the largest concentration of sucrose will have a larger concentration gradient, whereas the bag with just water will have an equal concentration gradient with water. So the procedure for this lab was we had to design our own experiment. We obtained 4 15mm dialysis tubing segments. You had to clip off one end of each bag using a clip, of course. Then you added 10 milliliters of deionized water to one bag. That would be the control for this test. Then you would add 10 milliliters of 10% sucrose solution to another bag. To find the correct ratio of water to solution, we had a 30% stock solution. You had to use this equation. So the original concentration is 30. So you have to find how much of that to get a solution of 10% in 10 milliliters. So 30 times x equals 10 times 10, so 30x equals 100. X equals around 3.3. So what we did is we had 3.3 milliliters of the 30% stock solution and 6.7 milliliters of water to get 10%. Next was you had to add 10 milliliters of 20% sucrose to another bag. Use the same formula. This time it's 30 times x times 200 equals 200, and you get x equals 6.7. So we added 6.7 milliliters of 30% stock solution and 3.3 milliliters of water in a small beaker and added that to the dialysis tube. And the final step, I think it's a little hard to see here, but you add 10 milliliters of 30% sucrose solution, which is the original stock solution, to the final dialysis bag. So continued, you clip off the other end of each bag. We had to weigh each bag first. Then you obtain 4 beakers filled with deionized water. You add one of the bags to each beaker. Each bag has its own beaker. And every 15 minutes, we weighed each bag. And we did 4 times for a total of 1 hour. And then after that, we decided to find the volume of each bag by emptying the contents into a graduated cylinder. Then we recorded our data and found the rate of water movement, which you'll see later in this presentation. So here are the results for weight. These are the different concentrations. And for 0%, as you can see, it starts it starts at a higher weight than 10%, 20%, which surprised me, but it follows the same pattern. Here it goes up, but notice it goes up very slowly. Like here, there's a change of only 0.01 gram in 15 minutes. Whereas 10%, it gradually rose at a greater rate. Same thing for 20%, and same thing for 30%. The general trend here is if you look down at the minute marker, and I'm sorry, earlier I said we weighed the bag 4 times, I think. We did weigh it 5, once at the beginning, and then every 15 minutes, 4 times. The general trend here, you can see it for 30, 45, and 60 minutes, is as the concentration goes up, the weight goes up. It applies here, here, and it does apply here. Here, it doesn't apply, well, generally, because this one has a difference, but this one increased more than this one, I believe, or just about the same. And in the end, the heaviest bag was the one with the 30% concentration. Okay, I paused and went back a second just to make a correction. These two here generally change the same in weight, but as you move along, this one gains more weight as the 0% concentration. So then we did volume, which again shows a general trend. As the concentration goes up, the larger the volume. The reason these two are the same is because obviously the 10% would be larger, but using the equipment that we used, using the equipment that we had, it was very hard to notice a difference in these two. So we had to record them as both being 10 milliliters after the hour was up. But there were noticeable changes in the 20%, 30%, especially the 30%. It gained 2.5 milliliters of solution. So the next experiment was finding the effect of osmosis on cells. And osmosis can cause cells to crenate, lyse, or stay at a normal state, which I stated earlier in the presentation. In animals, placing a cell in a hypotonic solution will cause it to lyse or burst. Animal cells have no cell wall, so it's easier to lyse. But in plants, there's a cell wall, which is very rigid, and it makes the cell much harder to break. So if a plant cell is placed in a hypotonic solution, it will create what's called turgor pressure. The cell will fill with water, but it won't lyse, and that's what makes plants stand up. So our hypothesis for this lab, this is the table we used to state what the solution was. And 0.09 would be hypotonic, 0.9 would be isotonic, and 10% would be hypertonic. Because the salt concentration of a red blood cell, in this case, is 0.9. And our hypothesis was that the hypotonic red blood cells would burst, the isotonic ones would look normal, or stay the same, and hypertonic would crenate. So the procedure for this lab was very simple. We viewed sheep's blood in the 3 concentrations at 400 times magnification and took a picture of each different scenario. So the results. Here's the blood at a 0.09% salt concentration. There are no red blood cells because they have lysed. Here are red blood cells at 0.9% sodium concentration, which is also the concentration of the red blood cells themselves, so the cells look generally normal. And here's the 10% salt concentration. As you can see, the cells that are here look shriveled or shrunken or crenated, which is exactly what they did. So our conclusion was, this is for the first experiment, that over time, the weight of the dialysis bags increased for all 4 concentrations. At the end, the weights for all 4 bags were larger. But as the concentrations increased, the weight also increased. So in the end, the heaviest bag was the 30% concentration of sucrose, but the lightest bag was the 0% concentration of sucrose, or the control. Also, generally, the volume increased as the concentration increased. And this happens because sucrose cannot move through the membrane, but water can. And water tries to create, and what it happens is it creates an equilibrium with the sucrose. The more sucrose, the more water you need to create an equilibrium. So the rates of water movement for the 0% sucrose concentration was around 0.01183 grams per minute. 10% was around 0.02350 grams per minute. 20% was around 0.04630 grams per minute. And 30% was 0.05817 grams per minute. As you can see, the trend is it was faster as the concentration was higher. And next was the sheep's blood. So the sheep's blood at 0.09 concentration contained no red blood cells, and that's because all the cells were placed in a hypotonic solution and the cells lysed because they had much more water than the solution. The sheep's blood at 0.9% looked normal, and that's because the concentration of sodium in the blood and the solution were equal. And the red blood cells in the 10% salt solution crenated, and this is because the concentration of sodium was hypertonic to the blood. And that's the end of this presentation. I can't really think of anything to say other than hopefully the Eagles don't choke again. So that's it. Thank you.